Hello, I'm Sean, and in this video we're going to explore the role of the cell cycle in determining cell fate. First, let's get a bit of motivation for this topic. In the late 20th century, it was well known that the cerebral cortex was constructed from the inside out. Progenitors that arose early in the development process became the deeper neurons, such as layer 6 neurons, and progenitors that arose later in the development process became the superficial neurons, such as layer 2 and 3 neurons. However, a general understanding of the underpinnings for this specific development pathway was not well understood until Susan McConnell's study in 1989. McConnell discovered that young progenitors that were transplanted to late development stage hosts would adopt the fate of its environment. For example, progenitors that would have normally become layer 6 neurons instead became layer 2 and 3 neurons. Moreover, McConnell found that this fate adoption relationship only applied in one direction. If late progenitors were transplanted to early development stage hosts, they would retain their old fate. For example, progenitors that would have normally become layer 2 and 3 neurons stayed as layer 2 and 3 neurons even after transplantation. Unfortunately, this discovery was not as simple as originally thought. McConnell and Kasnowski later discovered in 1991 that the young progenitors did not always adopt a late fate upon transplantation. Sometimes they kept their original fate. For example, progenitors that would have normally become layer 6 neurons sometimes became layer 2 and 3 neurons, and sometimes kept their original fate as layer 6 neurons. McConnell and Kasnowski realized that cells that were transplanted prior to the G2 phase would adopt the new fate, but those transplanted during and after G2 would be committed to their original fate. Before we delve further into the specifics regarding the relationship between the cell cycle and cell fate, we will first have a quick refresher on the cell cycle. In eukaryotic cells, the cell cycle is divided into two main processes: interphase and the mitotic phase. Interphase consists of the G1, S, and G2 phases. The mitotic phase consists of mitosis and cytokinesis. The end result of the mitotic phase is the creation of two new daughter cells from a parent cell. For the topic of cell fate, we will focus primarily on interphase. G1 phase, also known as the first gap phase, is primarily characterized by cell growth, organelle creation, and the construction of cellular implements necessary for DNA replication. S phase, also known as the synthesis phase, is when DNA replication occurs. G2 phase, or the second gap phase, is the final process of interphase. During this period, the cell continues to grow, create organelles, and begins preparations for mitosis. Once G2 ends, mitosis begins. Today, G1 is considered the most salient phase of the cell cycle in the process of deciding cell fate. It has been shown that cells in G1 will respond to specification signals more rapidly than cells at any other cell cycle position. G1 cells behave differently largely due to the specific changes that occur only during G1. Most notably, when cells transition from the mitotic phase to G1, the cell undergoes nuclear envelope reformation, chromosome decondensation, and chromosomal repositioning. Much of the DNA in non-dividing cells is often not permissive of transcription factors and other modifiers because it is bound into chromatin, a structure made primarily of DNA and histone proteins. However, during G1 and S phase, these chromatin structures become permissive, which ultimately allows for cellular specification modifications to be made, often manifesting itself in the addition of transcription factors. The reason why G1 is largely considered to be more salient than S phase for cell fate is because many cases of cellular fate determination is dependent on selective gene expression via transcription factors. The activity of such factors often peak in G1, which is why G1 is primarily emphasized in discussions revolving around cell fate. However, the ability to determine cell fate is primarily reliant on the ability to modify gene expression. Transcription factors are just a single means of achieving this. There are many other ways for cellular differentiation to occur, one of which even relies on making chromatin modifications for the expression of genes important for cell fate. For example, one method modifies histones, which are the proteins that DNA wraps around to create chromatin. By modifying these proteins and ultimately the structure of chromatin, gene expression can be induced or inhibited. As cell potency, or the possible number of cell fates, decreases, the length of the cell cycle will increase as well. For example, cell cycles in early development can be as brief as a few hours, but as more cell differentiation decisions are made, this time frame can increase to span days. During the development of the brain, it was shown that this cell cycle lengthening even had a causal contribution to neural progenitors switching from proliferative divisions to neuron-generating divisions. Overall, the cell cycle not only provides important information about the progression of cellular differentiation, but its specific phases and timeframes even play a pivotal role in the determination of cell fate.